All right, well, welcome to Neurocare Academy, and very happy to have Aron Hill with us today. Dr. Aron Hill from the Cognitive Neuroscience Unit at Deakin University in Melbourne, Australia. He's in Melbourne, a member of the ABSS as well, the Australian Brain Stimulation Society. Really looking forward to having you talk to us today, Aron, about your research. So welcome.
Thanks, Trevor. Thanks a lot. Thanks for having me. Should be fun.
Fantastic. Yeah, look, you guys are doing some great work down there in Melbourne, and look, really looking forward to hearing what you have to say about brain stimulation techniques, neuromodulation, how that's applied in the clinic, and hopefully this will be viewed by some people who are aspiring to do some similar sort of work that you're doing at the moment as well. So, thank you very much for being with us today. And look, we've got a few questions for you, some standard sort of questions that I've asked a lot of the people that I've interviewed so far. But look, I'd love you to start off with a little bit about your early years and how you got to what you're doing now, where you were born, where you completed your university education, that sort of thing. It's really nice for people who are aspiring researchers to know sort of the journey that you've been on. So yeah, how did that go for you?
Yeah, absolutely. Yeah, it's been an interesting journey, that's for sure. Yeah, so look, I grew up in Melbourne, Australia. So for anyone, I think most people are probably familiar where that is, but for anyone that's not, it's in the state of Victoria, down the bottom of Australia here, second largest city in Australia after Sydney and New South Wales. And when I went to university, I actually did, I guess, the broadest degree you could probably possibly do. I did a combined Bachelor of Arts and Bachelor of Science, because at that stage, I really wasn't sure exactly what I wanted to do. And I actually, as part of my science major, I actually majored in microbiology, because I thought that I might become a microbiologist. And I just took a psychology as my second major, just purely out of interest. I didn't actually think that I'd end up in the psychological sciences at all. But it turned out that actually, when I finished my undergrad, I actually worked as a microbiologist for about a year. And I realized very quickly it wasn't for me. I didn't like the sort of wet lab approach. I wanted to work with people. But luckily, I managed to apply for another job at the Alfred Hospital, which is a large teaching hospital here in Melbourne. And I got a position within the neurology unit there. And so I started working as a medical scientist. And so I was doing a lot of like routine clinical electrophysiological testing. So a lot of things like electroencephalography or EEG to help diagnose people with epilepsy and other brain disorders, and also a lot of nerve conduction studies and things. And sort of as I progressed, I started doing some sort of more complex work around things like small fiber neuropathies, movement disorders, work with Parkinson's disease, which I absolutely loved. I found it fascinating. And I guess the best thing about that as well was that I got to work with a lot of different people. I got to work with neurologists, neuropsychologists, and some various researchers as well. And although it was a clinical role, I got the chance to sort of get some travel grants and to go to a couple of international conferences. And that sort of really opened my eyes to what it was like to do research. And then I quickly realized that I think the research area was, what I was interested in. So following that, I went back and did my honours degree in psychology, and I actually did it at the Monash Alfred Psychiatry Research Centre. So this was co-led by Professor Paul Fitzgerald, who's well known in the brain stimulation field. And I did it using something called Transcranial Magnetic Stimulation, which I'm sure many people are very familiar with that are listening to this. But it's a technique where you place a coil over a person's head, and you deliver a very brief magnetic pulse. And that pulse is able to travel through the peripheral tissue, through the skull and through the scalp, and actually activate neurons in the cortex. And when that's applied over the motor cortex, it allows you to get a peripheral motor response called a motor evoked potential, which sort of gives you a marker, an indirect marker of cortical excitability. So as part of this project, what I did was I was looking at something called the human mirror neuron system. So mirror neurons are these neurons that have been found in several animals, including monkeys. that basically fire when you perform an action, but also when you watch someone else performing that same action. So for instance, if I were to grab reach out and grab a cup of coffee, some neurons in my motor and premotor cortex would become active. But if I were to watch you do that same action, the certain population of neurons called mirror neurons would also become active just by watching that. So this was sort of a chance to actually look at changes in cortical excitability when people watched people performing various actions, and that led to sort of a publication, which was really exciting. And then following that, sort of me wanting to go on and pursue a PhD.
Fantastic, Aaron. Look, you obviously got a lot of hands-on experience there, and a lot of exposure to real people, like you said at the start there, and that's, I guess, helped you to focus in on what, or to understand what you really enjoy is the research, and so then you went in to do some further research, and you went into to do a PhD. In fact, so can you tell us a little bit about the PhD?
Yeah, absolutely. So I ended up doing it at the Monash Alfred Psychiatry Research Centre, actually where I'd done my honours, but by this stage it had actually moved to a new lab just down the road from the Alfred Hospital. And I did it in a group called the Cognitive Therapeutics Group. Yeah, so I did it in the Cognitive Therapeutics Group, and this was a sort of a small group within the larger brain stimulation group at the MAPRC, and it was headed by Professor Kate Hoy, who's in the centre of this picture here. And the idea behind this group was really to use non-invasive brain stimulation techniques, particularly transcranial electrical stimulation, to modulate cognitive function. So the idea was that, particularly in a lot of neuropsychiatric disorders, people often have a number of issues with cognitive function and with memory function. And the idea was to try and use some of these devices to actually improve or enhance cognition using this. So that became the focus of my PhD work, where I was really interested in modulating cognition using these devices, and also sort of digging into the underlying neurobiology, sort of how were these effects occurring in the brain.
Fantastic. Yeah, look, a lot of psychologists and psychiatrists focus a lot on symptoms and to try to understand the underlying biological sort of etiology of these symptoms is really the major focus of neuromodulation, I guess, to try to get an understanding of why these phenomena are occurring in mental health. So it's great to have people like yourself who are looking at these biological factors. And so this can sort of help us to look at prognostically what sort of treatments can help certain symptoms. But look, that's fantastic, Aaron. So you went on to publish some of the research that you did from your PhD, and then after that, what would you consider sort of the top 3 sort of research publications, the stuff, things that you'd like to share with us today?
Yeah, sort of what I'll do is I'll give a little bit of background, just sort of so everyone's sort of familiar with the research that I've been doing, that will hopefully lead on to these publications. Sort of, as I mentioned, I was interested in transcranial electrical stimulation. In particular, I was interested in this technique called transcranial direct current stimulation. And this is a technique where, sort of, unlike TMS, which I was talking about earlier, which is able to stimulate the cortex and actually produce action potentials in neurons in the brain, this delivers much weaker electrical currents to the brain via 2 sort of sponge-based electrodes. There's an anode and there's a cathode, and the idea is that you connect these to a device that delivers a very small current, typically 1 to 2 milliamps, and the current travels from the anode to the cathode and just slightly alters the excitability of neurons in the brain. And the idea is that, sort of, under or around the anode, you get this increase in cortical excitability, and under the cathode, you get this decrease in cortical excitability. And I've just included here as well sort of an electric field model, sort of demonstrating that, where you can see when you place these electrodes, in this instance over the prefrontal cortex, you get this sort of electric field that occurs over that prefrontal region. And the thing that was interesting about this technique is it's not a new technique, but it sort of re-emerged in the early 2000s, and it became very, very popular. And I've just got a graph on the right hand side here, sort of showing some different brain stimulation techniques with TDCS in orange. And you can see that sort of from around 2005 and onwards, it's had this really big increase in the number of studies that have used this technique. And I think the reason for that is because it's got a lot of potential benefits, both as an experimental technique for understanding brain function, but also potentially as a neuromodulatory technique for potentially, being used therapeutically. These techniques are something that have been known for quite a long time. Back in the 1960s, there was actually some work done using electrical currents in animals, particularly in mice, and they sort of showed that depending on the type of current that you delivered, whether it was surface positive, which would be the equivalent to anodal stimulation, or surface negative, which would be like cathodal stimulation for TDCS, you could actually up or down regulate neural firing. And then sort of in 2000, there was a very seminal piece by Nietzsche and Paulus that showed using TMS very eloquently that you can either up or down regulate cortical excitability using TDCS applied over the motor cortex. But what I was really interested in, I guess, and sort of what my publications sort of stem from, is the idea of the effects on cognition. So there was a lot of original work that was done in the motor cortex using TDCS, but sort of around the time, sort of a few years before I started my PhD back in 2014, a lot of people started using TDCS in brain regions outside the motor cortex, particularly the prefrontal cortex and a part of the prefrontal cortex called the dorsolateral prefrontal cortex, or DLPFC. Because this is really important for a lot of sort of higher order executive functions, working memory, those sorts of things. And it's also often dysfunctional in neuropsychiatric disorders. And so there was a lot of research, really interesting research coming out showing that you could potentially modulate cognitive functions such as working memory by applying TDCS over these prefrontal regions. But it was quite a controversial area because there were a lot of conflicting results. There were a lot of quite small or underpowered studies. And there were a lot of studies coming out that showed these effects, but didn't actually show any underlying neurobiological mechanisms that might be occurring. So what I was really interested in was basically, first of all, can TDCS over the prefrontal cortex actually change or enhance cognitive function? And if so, how can it do it? What are the mechanisms that drive those changes? And so this sort of led to the first study that I did, which was actually a meta-analysis where sort of early on in my PhD, I basically took some studies and looked at the effects of anodal TDCS, which was typically used to enhance cognitive function, compared to sham, so placebo stimulation, on working memory performance. And I took all the studies that were done both in neurotypical, so healthy people, as well as in psychiatric populations. And sort of as a secondary aim, what I wanted to do is see if there were any dose response effects. So just to sort of basically look at things like current density and stimulation duration to see if they had any additional effect on the outcomes. So I ended up pooling a total of 352 subjects across 16 studies. There's obviously a lot more now, but this was done back in 2015, 2016, when there weren't a huge number of studies at that stage. And that was broken fairly evenly into individuals that were both healthy and or had a neuropsychiatric diagnosis. And sort of several key findings came out of this. What I found when I looked at working memory, I was looking at reaction time, so how quickly someone could respond to a working memory task, as well as their accuracy, how accurately they could perform the task. And I found that in healthy or neurotypical subjects, there was a small but a significant reduction in reaction time. So people's reaction times actually improved with TDCS, which was interesting. And when I broke that down into a finer subgroup, I looked at online and offline performance. So online is when someone's actually performing the task whilst they're receiving TDCS, whereas offline is typically they receive the TDCS and then they perform a task directly after it. And what I found is that there was no actual change in their performance on these working memory tasks whilst they were receiving stimulation, but following stimulation, so their offline performance, there was a small change. And similarly with accuracy, there wasn't a significant change, but there was this sort of, I guess, strong trend. There was a P of 0.05 suggesting that there might be a potential change in accuracy. But sort of conversely, in the psychiatric cohort, there was no change in reaction time for either the online or the offline tasks. And there was no change in accuracy, but when you broke it down into the subgroups, there was a significant moderate impact for online tasks, which was interesting. When I then looked at sort of these other stimulation parameters, sort of current density and stimulation duration, I probably should stress here as well that this was a fairly crude way of doing it. There weren't a lot of studies, so I sort of pulled all of the results together just to get a general idea of what was going on here. And I broke them down into sort of either lower or higher current densities, or lower and higher stimulation duration, sort of less than 10 minutes or more than 10 minutes of TDCS. And what I found is that accuracy was actually seen to be improved in the higher current density subgroup. So those who received more stimulation, a higher strength of stimulation. And also the longer stimulation durations resulted in faster reaction times. So this also indicated that there appeared to be some initial support for a potential dose-response relationship with TDCS, which was really interesting. But you know, I was very cautious with this because it was obviously modest effect sizes and not a huge amount of studies at the time. So I think there are a number of implications for this, and I think the first thing is that it did support some level of working memory enhancement with TDCS, which was good to know. But the effect sizes were pretty modest, and a lot of the studies or the measures actually failed to reach significance. So this really indicated to me that there was a clear need for refinement of stimulation. We needed to come up with more optimal stimulation parameters, or ways of delivering TDCS that might be more optimal and get more robust results. And I think sort of similar to that, there was a need for a better understanding of the physiological effects. So you know, if we could work out exactly the mechanisms that are occurring when we deliver stimulation, again, maybe then we'd be better at actually optimizing stimulation delivery. So that led to sort of the second and third studies that I'll just talk about very briefly here. With the second study, what I did is I had two key aims. What I wanted to look at was sort of really dig into the neurophysiological effects of TDCS over the prefrontal cortex, and also to see if the effects could be optimized. And by optimized, I meant using multi-channel or these so-called high-definition TDCS devices. So I sort of spoke about earlier that, you know, TDCS usually uses two fairly large sponge-based electrodes. There were these high-definition designs that use much smaller electrodes, and you can apply more of them. And they look actually more like EEG electrodes than TDCS electrodes. They're much smaller and they're gel-based. And you can place them around the head. And what you can do is you can create much more focal montages for more specifically targeting specific brain regions. The other thing that I used as well was something that I used and I continue to use now is something called TMSEG. So it's combined TMS with EEG. And essentially the reason for this is because I was obviously interested in assessing the neurophysiological effects outside of the motor cortex. So with TMS, you know, it's great on its own to measure motor cortical excitability, but obviously you need to get a peripheral motor response in order to get a motor evoked potential that you can record. The problem is if I wanted to look at the prefrontal cortex, I wouldn't get this motor evoked response. So I needed another way of collecting sort of some quantitative data. And TMSEG allows you to do this because what you essentially do, this is just a picture of Nigel Rogash, one of my former PhD students here demonstrating TMSEG, is you apply an EEG cap and you record EEG, and then you deliver TMS over the EEG cap. And what this allows you to do is it allows you to directly record the brain's response time-locked to the TMS stimulation. And the great thing about this is you get a response anywhere that you've got EEG electrodes. So you no longer need to just be looking at the primary motor cortex. It is a little bit tricky. It's quite a challenging technique to perform, and there are a lot of steps involved. And the output that you get is more complex. You get what's called a TMS evoked potential, or TEP, that I've shown here, which is a little bit more complex than an MEP in terms of analysis. We still don't know exactly what the various peaks represent, and that there's a lot of ongoing work to try and sort of delineate what these represent. But overall, it provides a good marker of cortical reactivity or cortical excitability. So what I did for my studies is basically embedded high definition TDCS electrodes within an EEG cap, and then applied TMS over the top of that. So there was a lot going on in the cap. It took a while to pilot this to get it to work. And I've just sort of got a cartoon diagram on the right-hand side, sort of showing the TMS coil over the high definition electrode with the TDCS electrodes surrounding it. But what it allowed me to do was to have everything there so that I could record brain responses before and after TDCS in the prefrontal cortex. So that's what I did. I basically did a 3-arm study where subjects came back 3 separate times, and they received either conventional TDCS over the prefrontal cortex, they received high definition TDCS, or they received sham stimulation. And I looked at the before and after effects of stimulation. And the other thing that I'll just touch on here as well is we also ran some electric field models to just look at current flow in the brain with these different montages. And sort of consistent with what we expected, you can see that the bottom high definition montage here produced much more focal electrical current within the prefrontal cortex. We were stimulating the left prefrontal cortex compared to the bipolar TDCS, indicating that it produced a more confined or restricted E field, which is what we were hoping for. So ultimately what we found is, and I'm just talking about some region of interest analysis here for the TMSEG, but we found that that both the bipolar, the active bipolar and the active high definition TDCS could actually increase cortical reactivity when we looked at TMSEG. We found specifically, though, there was a bit of a difference in response. So when we looked at the standard TDCS montages, what we found is it increased a specific component of the TMS evoked potential called the P60. And that occurred about 5 minutes after stimulation. Whereas when we looked at the high definition montage, what we found is there wasn't an effect until about 30 minutes after. It seemed to have this delayed effect. And when it did occur, it occurred more strongly. And this was really interesting because there'd been some previous work done by another group in the motor cortex using high definition montages that had shown using just conventional TMS that motor cortical excitability also has this delayed effect on motor cortical excitability. So it seemed to be what we were seeing here with our TMSEG results in the prefrontal cortex. So I guess the significance of this was really that it confirmed that we could have this plasticity inducing effect of TDCS in the prefrontal cortex. And this was pretty unique because no one had used TMSEG at this stage to look at the effects of TDCS over the prefrontal cortex. So this was able to confirm sort of results that I guess were somewhat consistent with the motor cortical studies, that we were also increasing plasticity in this region as well, which was pretty exciting. And that we also got some sort of differential effect between the 2 types of stimulation, whether we use the high definition TDCS or the standard. Exactly what that meant was unclear. And sort of, as I mentioned previously, we don't know a lot about the TMS evoked potential response yet and what the different peaks represent, particularly outside of the motor cortex. But one possibility is it was increasing cortical excitability. There's some evidence to suggest that the P60, which was modulated, might be a marker of cortical excitability. So if so, that would make sense because we know from the motor cortex that that's what TDCS appears to do, anodal TDCS. The final study that I did was sort of sort of flowed on from the previous one, and I used a similar setup. But what I did this time was actually look at what's called state dependency effects of TDCS. So the reason that I thought this was worthwhile is that there were typically 2 different ways that people delivered TDCS in these cognitive paradigms. Some people would deliver TDCS just while people sat and did nothing. So say you'd deliver 15 or 20 minutes of TDCS while people just sat on a couch with their eyes open or their eyes closed. Whereas some groups were delivering TDCS actually while people performed a specific task. So for instance, if you were wanting to say modulate working memory performance, what some people might do is actually get people to perform a working memory task while they were receiving stimulation. The idea being that you want to engage those circuits that are getting stimulated, and that might actually lead to this improved efficacy through these state dependency effects. So I wanted to sort of dig down into that in a little bit more detail. So again, I like with the previous study, I took a number of measures before and after stimulation. And what I looked at really, again, in a 3-arm study, a repeated measures design, is I looked at people receiving TDCS either while they remained at rest, people receiving TDCS while they performed a working memory task, or people receiving sham stimulation while they did a working memory task. And what I found when I looked at the TMSEG output was There was a reduction in the N100 amplitude of the TP for both groups, which was interesting because the N100 is potentially a marker of cortical inhibition, and this suggests that perhaps there was a reduction in cortical inhibition. But what was interesting is that the increase in P60, which we saw in the previous study, was there again, but it was only there when subjects remained at rest when they received stimulation. So this suggests that there was a bit of a differentiation between the type of plasticity that was produced depending on how TDCS was delivered. So sort of in summary, I guess, what we found overall is that there were some changes in these TP markers, particularly the P60 and the N100. And possibly they were most pronounced at later time points around 30 minutes post TDCS, as we indicated in the first study. So, there's a question around what this represents. I think one possibility is that it's a shift in excitation inhibition balance, because there's some evidence that the P60 might be a marker of excitability and the N100 possibly a marker of inhibition. But I think that's something that further work is definitely needed to sort of really dig down on and see if that is actually the case. But importantly as well, it suggests that these measures are sensitive to sort of these state dependency effects as well.
Well, that's brilliant. So many questions arise from those three studies that you've just talked about, Aaron. I'd love to sit here and discuss them for hours with you. One question that comes to mind is about the working memory application of TDCS or TES in general, and whether it can be applied in peak performance. You mentioned that there was a difference between the neurotypical population and the mental health population. So, one thing that we get a lot of interest in is peak performance. Do you think there's much application for it?
I think there is. I'm not super familiar with that area. But I think it's a really interesting area of investigation, because obviously there's a lot of people that are very interested in just trying to improve their cognitive function when they're already, healthy individuals. And there's a lot of people in particular wanting to improve things like sports performance and those sorts of things as well. I think it's an area that is going to see a lot more growth, but I can't comment specifically on how useful it will be. But I suspect that, again, I think it's one of these things with cognitive enhancement and with these devices in general. I think there's going to be more and more work looking at things like individualized approaches to delivering these. And when we understand sort of the key mechanisms that are involved, I think that will certainly give us a lot more opportunity to really sort of tease out these specific effects in specific individuals. Yeah.
Yeah, it's a great point. And I mean, the individual differences between people are paramount, and you showed that there was state dependency as well, so that when somebody was already activated, they possibly weren't showing the TEP or the P60, I think you mentioned, but at rest they possibly were. So an already activated brain may not show that that increase in activation from the TDCS, and you might find, or we might find, that with peak performance individuals, they might have a greater discrepancy between at rest and when they're activated. So all of these individual differences we're going to have to tease out before we can sort of, I guess, allocate different protocols to different people to get advantages, to get outcomes. Look, that's it's brilliant, Aaron, and you've done a lot of novel research there with new techniques and really pushing the boundaries of neuromodulation. So well done. We can't thank you enough for all the work you're doing, which just sort of pushes the field forward. It's fantastic. And so that's what you've done up until this point. What are you focusing on at the moment?
Yeah, so I focus on a couple of different things. I'll touch on briefly some of the work that I did overseas, and then come back to the work that I'm just starting now. Yeah, all right. So after I completed my PhD, I went overseas for a year to Canada, to the Centre for Addiction and Mental Health. So this is a large teaching psychiatric hospital in Toronto, in Canada. And there's a lab in there called the Temedy Centre for Therapeutic Brain Intervention. And that's a sort of a big, sort of well-known lab for doing a lot of non-invasive brain stimulation work in psychiatry, particularly TMSEG work, which I was particularly interested in. And what I did over there is I shifted gears a little bit. So I actually, they had some really some data on the use of convulsive therapies in people with severe depression. And I was really interested in looking again at the effects, sort of previous previously how I'd looked at the effects of TDCs with TMSEG and EEG, I was interested in looking at these effects using these convulsive therapies. To give a sort of a really quick background, I think most people are sort of somewhat familiar with electroconvulsive therapy, or ECT. It's something that's been around for a really long time. It's been around since the 1930s. And basically what it is there's a technique where you deliver electrical stimulation to the brain while someone's anesthetized and under neuromuscular blockade, actually induces a therapeutic seizure. So this is something that's used typically for people with really severe depression that isn't responding to other medications, what we call treatment-resistant depression. And it's actually incredibly effective. I mean, it has response rates of sort of somewhere between 50 to 70%, which is quite amazing. It's an incredibly useful technique, and it has a very rapid response as well, which can be particularly useful for people if they're acutely suicidal, for instance, and need treatment. But the problem with it is it has a lot of fear and stigma associated with it. You know, a lot of people associate it with movies like One Flew Over the Cuckoo's Nest and things like that. And you know, but in addition to that, it actually has a number of often quite severe cognitive side effects, particularly in relation to memory. So there's a problem with a lot of patients that might actually be getting treatment for depression and receiving benefits, but because they get these memory impairments, they actually might discontinue its use, which is a real problem. So there's a newer technique that is being trialed, and it came about, I think, in the early 2000s, and there was a lot of work being done in this group in Canada in particular, and it's called magnetic seizure therapy. So the idea with this technique is instead of delivering electrical currents, you actually deliver pulsed magnetic currents via a TMS device. It's essentially repetitive TMS, but using a specialized coil that delivers them at a really high intensity, strong enough to induce a seizure. And it's been found that this technique actually also has really good response rates. And also, really importantly, its cognitive profile appears to be much better than ECT. So it appears to have very minimal cognitive effects, which was really promising. So what I did sort of as part of this research, and some of the research that I'm still doing now in collaboration with the lab in Canada, is digging into the electrophysiological effects of these techniques and how they might be altering brain function. And to give an example of a recent study that I published, we looked at EEG connectivity before and after treatment with MST. And what we found is that after a course of MST, patients actually got increased connectivity within the theta band on their EEG. And they got, conversely, sort of this decrease in beta connectivity as well. And what we found is when we ran sort of some additional tests using receiver operating characteristic curves, or ROC curves, we could actually see that the change in theta connectivity actually was able to predict clinical response. So this was really interesting because it suggests that maybe, you know, this theta connectivity might potentially be some sort of biomarker that might actually be able to predict how people will respond to this treatment, which was really interesting. And in addition to that as well, we saw these sort of fairly pronounced changes in EEG spectral power, particularly in the lower frequency bands like the delta and theta bands, which were much higher than subjects when they started treatment, or healthy controls who obviously didn't actually receive any treatment at all. And similarly, we saw a reduction in gamma band as well. So really interesting to sort of see these different EEG effects of these convulsive therapies like MST. We also ran another study. This is one led by a postdoctoral researcher I worked with called Itai Hadas, and he also looked at the same cohort, but he used the TMSEG technique, and he did some fancy source localization and actually found that there were changes in connectivity and neural activity in several sort of subcortical brain regions in these subjects as well, which was really interesting. So particularly the anterior cingulate cortex, which is known to be involved in depression, and the hippocampus, which is obviously important for memory function. He saw these changes as well, which again suggests that it's having these fairly widespread effects on brain function, which was really, really interesting. Sort of more recently, sort of after I came back from Canada, I moved to the lab that I'm at now, which is the Cognitive Neuroscience Unit. So that's within the School of Psychology at Deakin University here in Melbourne. And this is it's a relatively new lab. It started in 2014, and It's led by Peter Endicott, who funnily enough was actually my honours supervisor years ago when I started doing my research. He moved over to this department, and the reason I joined here is because they do a lot of non-invasive brain stimulation, which is a fairly niche technique. You know, there's not a lot of labs that do it on a large scale, so that really appealed to me. But they have a focus on neurodevelopmental disorders in particular. So a lot of the work that I think I'll probably be doing sort of in the next few years is going to focus mainly on those, particularly things like autism spectrum disorder. And I think these brain stimulation techniques are going to be really useful for trying to understand some of the neurobiology around those disorders, particularly sort of the cognitive changes that occur in those disorders, which haven't been as well studied, which I'm hoping to do using things like TMSEG and theta burst stimulation as well.
Fantastic, Aaron. Look, that's awesome. And look, you mentioned a little bit about MST, and one question that came to mind as you were talking about that is that what are the main differences between ECT and MST? I noticed that in ECT, obviously, as we all know, the patient is anaesthetized. In MST, are they conscious?
No, so they're anaesthetized just like they are in MST. So it's a similar setup in terms of the muscular blockade and everything else, because it still produces a seizure. Yeah. The difference is the way the stimulation is delivered. So it uses magnetic fields, and a key benefit of that is when you apply electric fields at a really high intensity, like you do with ECT, what you find is when you look at where the electric field goes in the brain, it covers huge amounts of the brain. There are ways of delivering it that sort of minimize it a little bit, but still really both cortical and subcortical brain regions receive really large amounts of electrical stimulation. And that could be one of the reasons why it has a lot of cognitive side effects, because it's getting those deeper structures like the hippocampus and potentially causing problems in neural activity in those areas. With MST, you can control it a lot more because it's a different shaped, specially designed coil. Typically, it's applied sort of over the frontal cortex. Although it still produces relatively widespread changes, they're much more focal than you'd see with ECT, and they're also confined a lot more to the cortex, just because of the way that magnetic stimulation works. It doesn't penetrate as deeply as the electrical stimulation does. So we think that because it's more focal and because it's more confined to the cortical layers, it's possibly not causing these neurocognitive effects because of that reason. So it's really interesting, even though it's producing a seizure, and even though that seizure becomes generalized like it does in ECT, it's really interesting to see the different cognitive profile that's produced.
Yeah, excellent. And obviously, you know, a lot less stigma attached to the MST as well. So, look, you mentioned a little bit about your future aspirations there, about working within your developmental disorders and Peter Endicott and autism spectrum. Anything else on the cards in the future, or is it the main focus on that?
Look, I think like my ultimate aim is really just to discover sort of more effective treatments for both neurodevelopmental and psychiatric disorders. That's kind of the ultimate aim. I mean, you know, to take autism as an example, a lot of these disorders like autism, you know, they lack any good pharmacological treatments that can really deal with the core underlying symptoms. So, I mean, this goes for other disorders as well, like schizophrenia. So they can be particularly difficult to manage, because even if you manage some of the core symptoms, there's some other symptoms like the neurocognitive symptoms that are really difficult to control and can cause a lot of problems with the individual. So I think one thing that I'm noticing, and that a lot of people are obviously noticing as well, is that a sort of a one-size-fits-all to neuromodulation isn't going to be good enough. We need to have these more personalized approaches. And I think they need to sort of work with an individual's underlying neurobiology. So I'm talking about sort of personalized medicine here, I guess, but for brain stimulation. And I think this is going to be a really critical step sort of moving forward. But I mean, obviously it's one thing to say that and another thing to do it, because these things can be incredibly difficult to achieve. And I know a lot of people are working on these, but I sort of hope that, you know, I hope that over the coming years, we're able to delve a little deep into the physiology of these disorders and sort of focus on delineating some of these mechanisms. A particular sort of area, I guess, to give an example that I'm really interested in sort of moving forward, is this idea of excitation inhibition balance. So in a number of psychiatric disorders, particularly in autism, but also in schizophrenia, there's an idea that there's an aberrant excitation inhibition balance, that it's sort of been it's sort of been changed in some way. And there's a lot of techniques such as magnetic resonance spectroscopy, for instance, that can look at like measures of GABA and glutamate, which are markers of excitation and inhibition, as well as TMS in the motor cortex that does this as well, that show these irregularities. So I think, a really exciting area, and an area that I'm hoping to pursue, is to use these non-invasive brain stimulation techniques to modulate excitation inhibition balance. Because the great thing about this is we know that they can do that. We know that TDCs, for instance, or theta burst stimulation can up and down regulate excitation and alter inhibition. So it makes sense to apply these to these disorders where we know that that's disrupted, and potentially get it back to some level of equilibrium, which might potentially have a huge a therapeutic benefit.
Yeah, look, absolutely. And look, as you mentioned there, the underlying biology of these disorders and the underlying biology of the neuromodulation techniques allows us to sort of predict response as well. And if someone, for example, is increasing their theta and decreasing their beta connectivity after a few sessions, we can predict response and say, well, this is probably going to be a responder, and we'll continue. And that sort of personalized medicine approach is obviously going to allow us to have higher response and remission rates. So look, fantastic, Aaron. You mentioned that a lot of people are working on personalized medicine. It's an approach which is desired by everybody, I guess. What do you think the sort of timeline is we're working with? What do you think as a potential clinician looking at this, not just as a young researcher, but as a clinician looking at having researched for 20 or 30 years, having been a clinician for 20 or 30 years, for example, trying to now implement some neuromodulation into the clinic, what sort of things do you think they should focus on initially, and what sort of things may be happening in the field in the near future, in the next 10 years or so?
Yeah, it's a good question. I mean, again, I think this personalization approach is going to be really key. I think there's some really interesting things around newer sort of forms of plasticity inducing techniques. So for instance, theta burst stimulation, a repetitive TMS technique, is showing a lot of promise. It's obviously it's recently been approved by the FDA in the US for the treatment of depression, and a really important thing with this is that it's dramatically cutting down the time it takes to deliver this this therapeutic benefit. So for instance, a typical repetitive TMS depression treatment might take 30 or 40 minutes to deliver. Well, with theta burst stimulation, we can do that in 3 minutes. So that's really exciting, because it means that it's going to be cut down. And it also means that potentially we can accelerate treatments. There's been some work by some groups. For instance, I know there's a group in Stanford that's looked at this, that's looked at applying a lot of different, separate theta burst stimulation protocols in a very short duration. So that these sort of accelerated protocols that seem to have this really good effect. So I think that's one example where it might become clinically useful, because if we can fit in more stimulation into a shorter time period, we might be able to get these more robust effects. And I think that's something that needs a lot more research still, but it's a really exciting avenue potentially. I think a large percentage of past research sort of on plasticity inducing non-invasive brain stimulation techniques is also focused on depression. So naturally, that's where a lot of this innovation has occurred. But I think there's no reason that this can't occur in other areas as well. I think we just need to sort of simply find, do the research to find the best approaches for generating benefits. And I don't think it's going to be a one size fits all approach at all. I think that, we really need to tailor this to the specific disorder and even the specific individual that's being treated. I guess the other thing as well that I think that is going to be something that's going to continue down the track and become even more important are these sort of multimodal approaches. So for instance, the combination of structural and functional neuroimaging techniques. I mean, we use structural MRI now to guide very precisely the coil placement for TMS, which has been shown to be very useful. But there's some really interesting studies showing that you can use functionally guided TMS as well. So for instance, the studies showing that you can take resting state functional MRI and you can work out these functional targets. And if you deliver stimulation over those targets, you might actually get a better response. Because I think we're learning more and more that many sort of neuropsychiatric disorders are the result of these dysfunctional neural networks rather than just being a specific brain region, which was, you know, common to think of 10 or 20 years ago. So I think that these like circuit-based or functionally based network-based approaches are going to be really key to help improve efficacy of these techniques as well. I think finally as well, just for TDCS and transcranial electrical stimulation, I think a key benefit is really like affordability and portability. I mean, that was why I was really interested in these techniques, because you know they're easy to get out to a lot of people, and I think again take home methods of particularly TDCS and TES are going to be really important, because if you get severely ill patients, for instance, with depression, it might be very hard for them or very difficult for them to come into to get treatment every day. But if you could actually give them a TDCS device to take home, and they can safely administer that themselves, then that might actually be hugely beneficial as well, because then they can just administer the stimulation every day and potentially save a lot of time and a lot of hassle as well.
Yeah, look, I think you raised some very important points there. I think the accessibility to this treatment, the price of it has come down, and we can actually use these interventions in small clinics and even remotely in big countries like Australia, where a lot of people don't have access to the clinic. But I think one of the limitations that we have at the moment as well is that the assessment that we need to do using fMRI example, requires. I mean, it's a costly assessment, and to try to bring this into the clinic using things like EEG. I mean, you showed a little bit about EEG and source localization. That sort of thing could quite possibly be done in a smaller clinic. It requires the person to come in for the assessment. But look, you raised some great points, and we're moving towards being able to do everything in a smaller clinic. But the assessment part is obviously a major part in personalizing the intervention after that, which then becomes cheaper and more portable. So look, that's awesome. Thanks, Aaron. Look, I've got one last question very quickly. You've already mentioned some of the advantages of TDCS and TMS, but in the clinical practice, for somebody who's there right now in today, what sort of things, in what sort of situations should they start to try to implement neuromodulation? What are the advantages in comparison to other therapies that are currently being used?
Yeah, a good question. And I think for me, sort of one of the most exciting aspects of it, and the reason that I'm really interested in brain stimulation, is basically just the ability to directly target dysfunctional brain regions or circuits. So by directly stimulating the brain, we have this sort of direct window into potentially a therapeutic option. And with many drug-based interventions, they don't have this precision, unfortunately. I mean, it can be difficult to design a drug that selectively targets a very specific neural process. So although there's obviously a lot of pharmacotherapies out there that might be quite effective, a lot of them have unwanted side effects. And a lot of that is because they're not incredibly selective at targeting what we want them to target. So I think if we're able to sort of continue to optimize these brain stimulation techniques to improve selectivity. So for example, through some of the things I've talked about, like through a combination of using, precision neuronavigation or using state dependency or personalized stimulation techniques, which we can do now, and which I'm sure will just improve in the years to come, we can probably get improved therapeutic effects. With hopefully, I mean, ultimately what we hope is to get, robust therapeutic effects with very little side effects. But I mean, obviously that's what everyone's hoping for, and that's a work in progress. I guess sort of finally, I guess another key benefit is, yeah, again, that I keep touching on, is just the potential for personalization. I mean, if we're able to adapt certain stimulation parameters based on an individual's neurobiology or their neuroanatomy, I mean, this could obviously be incredibly advantageous as well. And it could be something as simple as, titrating TMS or TDCS dose based, for instance, on the electric field models. And if we can work out the right amount of stimulation to provide, then possibly that's going to be incredibly beneficial and could be a relatively easy thing to do as well, and isn't too expensive.
Yeah, look, fantastic. And hopefully, we can get this sort of information out to everybody as quickly as possible so that we can, care for individuals a lot better and more effectively as soon as possible. So thank you very much for your time. You're obviously right on top of all of the literature and all the newest techniques, and love to have you back again another time, perhaps in a year or two, get an update on what you're working on and where the field's at. But look, thank you very much for your time, and I wish you all the best for everything in the future.
Thanks, Trevor. I'd love to. Appreciate it.
Okay, great. Thanks, Aaron, and all the best with everything. Cheers.
Thank you.
Bye.